Hello everyone, my name is Sumit. I am back with the new video. Today we will discuss about the nucleic acids and the enzyme. Nucleic acid are long chain polymeric molecules. The monomer or the repeating unit is known as the nucleotides, and hence sometimes nucleic acids are referred to as polynucleotides. Nucleic acid can be defined as organic molecules present in the living cells. It plays a key factor in transferring genetic information from one generation to the next. Nucleic acid are composed of DNA, deoxyribonucleic acid, and RNA ribonucleic acid that form the polymers of nucleotides. In the nucleus, nucleotide monomer are linked together comprising of distinct components, namely a phosphate group, nitrogenous bases or ribose and deoxyriboses. Pyrimidines and the purines are two types of nitrogenous bases. Pyrimidines are composed of cytosine and thymine. Purines are composed of guanine and adenine. Thymine is replaced by uracil in ribonucleic acid, whereas deoxyribonucleic acid comprises all four bases. Let's talk about the DNA structure. DNA consists of instructions that monitor the performance of all cell functions. It is a cellular molecule that is organized into chromosomes. They are present in the nucleus of the cells and contain the cellular activities. DNA consists of so many functions and the activities. It is a double helix formed by a two polynucleotide chain that are twisted. There are two strands of DNA which are parallel to each other. Hydrogen bond binds to helix and the bases are bundled with the helix. Due to the presence of phosphate group, DNA is negatively charged. Chemically, DNA is composed of a pentose sugar, phosphoric acid, and some cyclic bases containing nitrogen. The sugar moiety present in the DNA molecules is beta D2 deoxyribose. The cyclic bases that have nitrogen in them are adenine, guanine, cytosine, and thymine. These bases and their arrangement in the molecules of DNA play an important role in the storage of information from one generation to the next one. Then we will talk about the RNA structure. RNA plays a vital role in the synthesis of proteins that mainly involves decoding and translation of a genetic code and transcription to produce proteins. RNA molecules are also composed of phosphoric acid, a pentose sugar, and some cyclic bases containing nitrogen. RNA has beta D sugar and some cyclic bases containing nitrogen group in it as the sugar moiety. The heterocyclic bases present in the RNA are adenine, guanine, cytosine, and uracil. In RNA, the fourth base is different from that of DNA. The RNA generally consists of single strand which sometimes fold back. Then we will talk about there are several different types of RNA and each has a specific function. Ribosomal RNA, it is one of the components of ribosome that are involved in the protein synthesis. Then transfer RNA, it is essential for the translation of mRNA in a protein synthesis. Then microRNAs, it is the smallest among of all RNA that helps in the regulating gene expression. Then messenger RNA, it is the RNA transcript that is produced during DNA transcription. Then function of the nucleic acid. Nucleic acid is responsible for the synthesis of proteins in our body. RNA is a vital component of protein synthesis. Loss of DNA content in link to many diseases. DNA is essential component required for the transferring genes from parents to the offspring. All the information of cell is stored in the DNA. DNA fingerprinting is a method used by forensic expert to determine paternity. It is also used for the identification of criminals. It also plays a major role in the studies regarding biological evolution and the genetics. Then next is enzyme. Enzyme is the majority of enzyme are proteins with the catalytic capabilities crucial to perform different processes, metabolic processes, and the other chemicals reaction in the cell are carried out by the set of the enzyme that are necessary to sustain the lives. Then we will talk about the function of the enzymes. The enzymes perform a number of functions in our bodies. Enzymes help in a signal transduction. The most common enzyme used in the process includes the protein kinase that catalyzes the phosphorylation of the proteins. They break down the large molecules into a smaller substances that can be easily absorbed by the body. They help in the generating energy in the body. ATP synthase is an enzyme involved in the synthesis of energy. Enzymes are responsible for the movement of ions across the plasma membrane. Enzymes perform a number of biological reactions including oxidation, reduction, hydrolysis, etc. to eliminate the nutritive substances from our body. They function to recognize the internal structure of the cell to regulate the cellular activities. Then we will talk about the enzyme classification. In that, first one is oxidoreductase. This catalyzes oxidation and the reduction reaction. Example, pyruvate dehydrogenase catalyzing the oxidation of pyruvate to acetyl coenzyme A. Then next is transferase. This catalyzes transferring of the chemical group from one to another compound. An example is a transaminase, which is transfer an amino group from one molecule to another molecule. Then next is hydrolysis. This catalyzes the hydrolysis of a bond. For example, the enzyme pepsin hydrolyzes peptide bond in a protein. Then next is lysis. This catalyzes the breakage of bonds without catalysis. Example, aldolase, an enzyme in glycolysis, catalyzes the splitting of fructose 1 6-biophosphate to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Then next classification is isomerase. They catalyzes the formation of an isomer of a compound. Example, phosphoglucomutase catalyzes the conversion of glucose 1 phosphate to glucose 6 phosphate. Is transferred from one to another position in the same compound. In glycolysis, glycogen is converted into the glucose for the energy to be released quickly. Then next is ligase. Ligase catalyzes the association of two molecules. For example, DNA ligase catalyzes the joining of two fragments of DNA by forming a phosphodiester bonds. Then next is cofactor. Cofactors are a non-proteinous substance that associated with the enzyme. A cofactor is essential for the functioning of an enzyme. The protein part of a enzyme in cofactor is apoenzyme, and enzyme and its cofactor together constitute the holoenzyme. This is all about the nucleic acid and the enzyme. Thank you.